Oi, catch.
Ah! Whoa, what are you doing? I can't leave you two to do anything. This is an onion. Inside this onion are loads of cells. The problem is we can't see them with our naked eye. So what we have to do is we have to use something called a microscope. Now, in order to see these cells, we're going to have to take an onion cell slide, and that is what this practical is about today. This is the core practical for onion cell slides. Within this core practical, there is set equipment that we're going to use. The equipment that we're going to have today is iodine, forceps, a scalpel, a tile, an onion obviously, we're going to have a microscope slide, and then we're going to have cover slips. And obviously the most important bit of equipment that we've got for today is the microscope itself. Now we're going to talk through how the microscope works, and then we're going to do our experiment. So what we have in front of us is the microscope. Now the microscope is made up of lots of different bits, but the most important bit of it is the light. And that is because this is a light microscope. So the reason that we can see cells through this microscope is because light is shone from the bottom here all the way through into the eyepiece, into our eye. And so because of that, we're able to see the microscope slide itself. Now, this eyepiece here, which I can remove, has got a lens inside. And that lens increases the size of everything it sees underneath it by 10. So that is a 10 times magnification eyepiece. Now at this point I'm going to pick up the microscope. Now whenever you pick up a microscope, it's really important you hold it by 2 points. The first point is on the arm and the second point is on the base. That gives it stability when you're walking along with it. Now the reason I'm turning it this way is so that you can see these lenses. The lenses here each have their own lens in. And so what we have is, we have this one here, which has got a lens that makes everything 4 times bigger. We have this lens in the middle, which makes everything 10 times bigger. And then we have a lens here that makes everything 40 times bigger. Now at this point, we can calculate the overall magnification. And the way that we do that is we say, well, this eyepiece was 10 times bigger and this first lens was 4 times bigger. So what we do is 10 times 4. Now that means we have a magnification of 40 times bigger. Therefore, when we turn it to the 10 times bigger, 10 times 10 is 100 times bigger, and then 40 times 10 is 400 times bigger. Now that enables us to see the cells much more clearly, because obviously if I was made 40 times bigger or 400 times bigger, then I would be massive. Whoa, 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 I didn't say to make me bigger, I was just saying the cells would get bigger. So I'm now going to move it round, and what you can see here is the stage. The reason it's called the stage is a bit like the reason we have stages for actors. It's because this is where all the action is going to take place. We're going to place our microscope slide here. You then have these stage clips which hold the microscope slide in place. Now, as if by magic, here's one I prepared earlier. And so what I'm going to do is place this on the stage, and then I'm going to hold it in place like so. Now, at this point, I'm now going to want to see if I can find the cells on this microscope slide. So at this point, I'm going to turn this round and I'm going to look through the eyepiece. Now, it's really important when you look through the eyepiece that you make sure that these lenses are in position. And the one you want to start with is the smallest lens, so the ×4 lens, which is going to make everything 40 times bigger when the eyepiece is added. So I turn that, and then what I do is I look for the cells. So we're now ready to do the practical. First thing we need is the onion and the scalpel. So the first thing I'm going to do is cut the onion so that I can remove a section to remove some of the cells. So what I've now got is my section of onion, and I'm going to peel using the forceps, and they are pretty much tweezers. I'm going to pull off a section of onion skin. Now what's really important, and this is more important than anything else you do in this practical, is you need to make sure that that section of skin is 1 cell thick. You cannot see the onion cells without it being that thin. And the reason is because otherwise you'd have layers of cells on top of each other, which would mean that you wouldn't see the detail of an individual cell. So what I'm now going to do is, as I remove this section of skin, I'm going to place it onto the microscope slide like so. And what I'm going to need to do is make sure that it's laid perfectly flat. The reason is that that will then prevent any bubbles underneath or air gaps, which will make it more difficult to see the cells. So once that is flat onto the slide, we will now add the iodine. Okay, so I've now got my iodine. What I'm going to take is just 1 drop onto the onion cell slide. And at this point, I'm now going to need to add a cover slip. Now, this is the bit that's really important. The cover slip is really, really, really thin. It's made of glass. It's incredibly delicate. And so what you're going to have to do is angle the cover slip so that it's at about 45 degrees. And what will then happen is you will lower that down and then you'll let go and the iodine will spread out over the cell slide. It's really important that if you get any iodine run through, you get a piece of filter paper and you draw through any excess iodine that's there. Now, the reason we use iodine is because it's a stain. Without the iodine present, you would not be able to see the cells in detail. So the iodine makes parts of the cell show when you're looking at it under a microscope. And that's exactly what we're going to do with this now. We're going to look at it under a microscope. So I've placed the microscope slide onto the stage. I've put the clips on top to hold it in place. And so what I'm now going to do is I'm going to look through the eyepiece and try and focus the lens so that I can see the cells in detail. So what I'm going to do now is I'm going to use the focusing wheels to try and get the onion cells into focus. Now I turn it very slightly until I can see the cells coming into focus, and I keep turning until they're all completely in focus. The moment they are, then I can begin to use the fine focusing wheel for any greater detail. So what you can see here is that you've got loads of cells. We can see more than we could do with the naked eye because we obviously couldn't see anything then, but we still can't see enough detail. So at the moment these cells are 40 times bigger, and look how tiny they are. They're really, really tiny. So what we're going to do now is we're going to make it 100 times bigger and see if that means we can see more of what's inside the cell. Well, that's bigger, but it's still not big enough really to be able to make a cell drawing. So at this point you can see the cells are a bit bigger, you can see a bit more detail, but we want to see more. So what we're going to do now is make it 400 times bigger. So now we've got the whole cell in view. Now what's really important at this point is that we begin to look for structures within that cell. So what we're looking for is the nucleus. So what we can see is we've got a nucleus in shot, we can see the cell wall, we can see that cell wall is then joined to other cell walls, and you can begin to see the arrangement of how those cells are all packed together. You can see then how the plant then gets its structure with these strong solid cell walls. So at 400 times bigger, I can now do my drawing of my plant cell that I want to do, because it's got all the detail in place and I only really want to draw 2 of them. So at this point, I'm going to hand back to me. So now I've got it in focus at the times 400, I'm now going to do a drawing of what I can see under the microscope. Now what's really important with this is that you only draw 1 or 2 cells. It's not designed for you to draw loads and loads and loads of them, which is why we're doing it at times 400, but we're going to try and get as much detail in as what we can see. I'm going to use a really sharp pencil. I'm going to make sure that it's one continuous line that I'm drawing and it's no shading. So hopefully from today's practical, you will have taken away that we use a stain in order to be able to see the cells more clearly. We use an eyepiece that has its own magnification, and then any lens we use after that, we have to times the eyepiece magnification by, and that hopefully you've understood how to make a cell slide.